Okay, well, good afternoon, everybody. I think we'll get started with this webinar for the Basic and Translational Science Working Group. I'm Sanjay Sinha. I'm at the University of Cambridge in the UK, and I'm delighted to bring and host and chair this webinar on the use of induced pluripotent stem cells for modeling aortic diseases. We've got two absolutely great talks today, and I think this is going to be a very informative webinar. It's an area that is cutting edge, it's exciting, and I think it has a lot of potential to really inform patient management. So I'm going to introduce you to our speakers. We have two speakers today. First of all, Dr. Hongorzul Davaapil, who is a postdoctoral research associate at the University of Cambridge. Hongorzul, she did her PhD at University College London, where she looked at the effects of basement membrane proteins on blood-brain barrier integrity. She then did a postdoc in my lab, or she's doing a postdoc in my lab at the University of Cambridge, where she's using iPS cells to model Marfan syndrome. And in particular, she's developing the use of these cells in this model system for drug screening and for 3D modeling, with the aim to accelerate the discovery of new therapies for patients. Our second speaker is Dr. Dogukan Mizrak, who is at the University of Michigan. So he did his PhD at New York University, and then he obtained a postdoctoral fellowship at Columbia University, where he worked on stem cell biology and next generation RNA sequencing. In 2020, he obtained a position in the Department of Cardiac Surgery at the University of Michigan as a research assistant professor. And he's working in collaboration with Dr. Bo Yang, combining human-induced pluripotent stem cells for gene editing, tissue-engineered blood vessels, functional genomics, and to establish more effective platforms for aortic aneurysm treatment development. So we'll move on to the questions to the talk in just a minute. What I wanted just to say is, if you have any questions, please type them into the Q&A box in Zoom. We'll have both speakers give their talks, and then we'll have the sessions at the end in a panel sort of format. So with that, I will ask the first speaker, Hongorzul, to start.
Great. Thank you very much, Sanjay.
I think I just need to stop sharing. Right.
Great. All right. Can you see my screen?
Yes.
Yes,
Yes, we can.
So thank you very much for that introduction. My name is Hongorzul, and I'm a postdoc in Professor Sinha's group. And the talk I'll be giving today is to go over modeling genetic aortopathies using stem cells, the advances, the challenges, and future outlooks for this field. So thoracic aortic aneurysms are, of course, a condition where there is an enlargement of the aorta, which have consequences for the patient that are quite fatal. So these can lead to dissection of the aorta or rupture of the aorta, as well as potential need for elective surgical repair, which is in of itself quite high risk. Unfortunately for patients, the treatment options are quite limited, and it makes the management of these diseases to be very tricky. Of the syndromic diseases which have a component of aortic aneurysm, we have, of course, Marfan syndrome, Loeys-Dietz syndrome, and vascular Ehlers-Danlos syndrome. And for the purpose of my talk today, I'll be focusing in on Marfan syndrome. So identifying new novel therapies for Marfan syndrome has been quite tricky. So over a decade ago, there was some really wonderful work that showed that treatment of a Marfan mouse model using losartan, which is an angiotensin receptor blocker, led to really wonderful rescue of the aortic phenotype in these animals. And this led to a lot of excitement and a number of international clinical trials across the world, where losartan was then used in treating patients with Marfan syndrome. And unfortunately, the outcomes of the clinical trials were not as clear-cut as for the mouse model, where you had some patients that responded quite well to treatment and other patients that didn't respond very well to treatment. And I think there are a number of reasons why this could be the case. I think some of the more obvious ones could be things like species-specific differences, could be considering things like the drug response for mice could be much higher than what perhaps patients would be able to tolerate in the long run. But I think there's also a component of the fact that in a mouse model, you are working with one mutation, one causal mutation in fibrillin and one genetic background, whereas actually in patients, you have thousands of potential mutations in fibrillin and a huge diversity of genetic backgrounds, which might have a role in dictating how disease progresses. So this then kind of leads us to some key questions of how do we overcome patient heterogeneity? Because this is clearly an issue that we have to address if we want to identify good treatments for patients. Is there a way that we can predict drug responses in patients? And is there a way where we can discover new and novel treatments for them? And to address these issues, our lab, we employ the use of induced pluripotent stem cells, or iPS cells. And these are stem cells which can be derived from human patients themselves. So these are basically fibroblasts or PBMCs that are reprogrammed into stem cells. And of course, the stem cells themselves can then be differentiated towards a variety of cell types, including vascular muscle cells, which we think is one of the major players in aortic aneurysm formation. So I'm going to give you an overview of my talk. So I'm going to first talk about some of the applications of iPS models, just basic introduction of the model itself, talk about some of the phenotypes of Marfan iPS-derived vascular muscle cells, and some of the more recent work with regards to drug screening before going into some of the kind of more technical issues, such as reproducibility and the cost of iPS research. So the vascular muscle cells that make up the aorta are derived from different embryonic origins. So the cells at the aortic root are derived from the lateral mesoderm or secondary heart field. The cells that are at the ascending aorta or the aortic arch are derived from the venal crest, and they are derived from the paraxial mesoderm for the descending aorta. And we hypothesized that vascular muscle cell lineage may play a key role in kind of disease susceptibility of those particular regions, as we often find that aortic aneurysms form in very specific places in a lot of these syndromic diseases. So in the lab, we have an iPS model which we tried to recapitulate some of these embryonic specific origins. So we first create these cells by first generating the embryonic intermediate. So for example, if you are interested in looking at the aortic root, you may wish to first generate a lateral mesoderm before we then push them towards a vascular muscle cell fate. And this is a model which I think has a number of good attributes. So it's of course a human model. It is a model that is patient-specific, so therefore captures the effect of the causal mutation as well as the genetic background. And this is also a model which allows you to study the specific effects of lineages on disease, which I think is something that's quite interesting, quite important as well. It also has the advantage of allowing us to study earlier stages of disease, because of course, samples which we obtain from human biopsies are really kind of end-stage disease where repair of the aorta was very urgent and very necessary. You also get an unlimited supply of vascular muscle cells, which is also very, very good because obtaining good control healthy cells from surgeries can be very tricky because oftentimes if you have surgery, you often have a problem with your aorta. And the whole pipeline to go from iPS cell to mature vascular muscle cell takes approximately 1 month. So it's not short, but it's also not that long. So if you do want to do lots of assays, it's a system that is very much amenable to that. So I just want to kind of give you a bit of an overview of some work that's already been published. So this is work that's done by Alex Granat, and this was published in Nature Genetics back in 2017. And this is where she modeled Marfan syndrome using this system of iPS cells, where she took iPS cells from a patient with a mutation in fibrillin, which resulted in a C1242 mutation. And the resulting vascular muscle cells were really abnormal. They had a number of issues, including increased apoptosis, increased proteolysis, abnormal response to mechanical stretch, and quite a lot of ECM abnormalities as well. And I think the quite nice thing about having this kind of system is the fact that it's very amenable to gene editing. So using CRISPR-Cas9, she was able to correct the mutation in fibrillin. So you end up with 2 cell lines that are virtually identical, apart from that one causal mutation in fibrillin. And the resulting cell line, as genetic control, was a much more normal cell line, and a lot of the abnormalities we see with the patient line are very much rescued. I also thought that it was quite interesting to point out that in this model, there were a number of lineage-specific differences that we can observe. So this, for example, is a panel of fibrillin-1 staining from the cells from control line as well as the patient line derived from the 3 different origins. We have lateral mesoderm, paraxial mesoderm, and neurocrest-derived smooth muscle cells. And you can see that while there are defects in the fibrillin deposition in the Marfan cells in all 3 lineages, those defects are the most pronounced in the neurocrest lineage, very much indicating that there may be kind of very lineage-specific effects at play and how disease may progress. So some more recent work that we've done is some drug screening that we have done in conjunction with AstraZeneca. So we were keen to try to identify new compounds which may improve the disease phenotype in vitro. So we knew that the cells were highly proteolytic. So we used this fluorescently quenched gelatin, which when it was degraded by the proteases, would result in a green fluorescence, which we can then measure and quantify. So we started off with 1,000 compounds, and those 1,000 compounds then got whittled down to 36 compounds, which were the ones which were able to reduce the proteolytic phenotype of the cells to levels comparable to the isogenic control. And from these 36 compounds, there were a total of 902 unique drug targets. So you have to remember that a single compound can have multiple enzymes that it can actually inhibit or target. So we did a lot of filtering based on affinity, based on kind of how recurring they were, and we came up with a list of 27 recurring drug targets. And this is just a small selection of these recurring drug targets. And you can see there are quite a lot of interesting hits to potentially study in the future. But the one which caught our eye was the one I've highlighted here, which was GSK3 beta. And this is the one that had the highest average affinity of all the drug targets from the compounds we identified. So we decided to look further into GSK3 beta as an interesting target to validate in our system. We verified some of the experiments that we did from the drug screen. We did a proteolysis assay, which was done in a very similar way to the drug screen. And we can see that without any drug treatment, the DMS or control, the Marfan cells were much more proteolytic compared to the isogenic control corrected cells. We then treated the cells with 6 different GSK3 beta inhibitors and found that treatment with these inhibitors resulted in a decrease in the proteolytic activity of the cells. In addition, we were also quite keen to do a genetic intervention as well, because of course we just discussed how a single compound might have multiple drug targets. We wanted to ensure that what we were looking at was not a result of some kind of off-target effect. And we had a look at knocking down GSK3 beta expression using siRNA and found that this was also quite effective at reducing the proteolytic phenotype of Marfan cells. We then had a look at apoptosis using a tunnel stain. And again, without any treatment, the Marfan cells were much more apoptotic compared to the control cells. And treatment with the GSK3 compounds resulted, most of all, in a decrease in the extent of apoptosis in the cells. The same thing can be seen when we looked at knockdown of GSK3 beta using siRNA, where we saw a nice decrease in apoptotic features of the cells. We were then interested in looking at how GSK3 beta might modulate the phenotype in additional lines, because the drug screening and some of the validation we've done was only done in one Marfan cell line. So we wanted to see what happens in other cell lines. So we had additional lines we obtained from Dr. Anchild, and these were severe patient lines. So we have highlighted them here. They're DE35, DE37, and DE119. And we noted that when we treat them with the GSK3 beta inhibitors, you can also decrease the extent of proteolysis and apoptosis in a very similar way you could with the original Marfan CU1242 cell line. But I think the most interesting thing to come out of this is the fact that when we repeated these experiments, but we treated a different cohort of cell lines, These were cell lines derived from patients with mild and moderate disease rather than severe disease. And we noted that there was a very different response to drug treatment. We noted that the cells do not have the same decrease in the proteolysis and apoptosis compared to the severe cell lines. And I think this is something that is very interesting, and it kind of mirrors what we were talking at the beginning about the fact that patients can have very variable responses to drugs. And it definitely seems in this case that GSK3 beta inhibitors seems to work, at least in vitro, in some cell lines, but not in other cell lines. So I'd now like to move on to some of the kind of technical drawbacks and considerations for iPS research, the problem of reproducibility, as well as the cost of iPS research. So I'd like to give you an example of kind of how reproducibility and variability is an issue in iPS research in general, not necessarily just in our field. And this is a paper which I think illustrates this quite nicely. It's published in 2018 in Stem Cell Reports. And this is where one single standard protocol was given to 5 different labs, and they were trying to generate cortical neurons. And I think you don't have to be a neurobiologist to appreciate the fact that they seem to have produced 5 quite different cells in this process. And this is something that is, of course, very problematic, because if you were then to try to collaborate with somebody, this can make things quite challenging. And they did some bulk RNA sequencing on their samples, and this is just kind of the PCA plot down here to kind of summarize a lot of their findings. And you can see that some of the labs were quite successful at making very reproducible neurons, whereas other labs perhaps had a much higher spread of the kind of products that they made. And this is something that's clearly going to be an issue, or going to have an impact if you're then using these cells for disease modeling in the future. So this is an example that's maybe a bit closer to home. These are, of course, vascular smooth muscle cells. And these are cells that I myself differentiated. This is a wild-type line, and these are neurocrest vascular smooth muscle cells. And here you just see staining for transgelin and calponin. These are vascular smooth muscle cell markers. And you can see that in the merged picture, pretty much all of the cells are positive for transgelin, and most of the cells are positive for calponin. And this is kind of what you would expect. And this is kind of a nice example of a differentiation that's gone quite well. About a month later, I did another differentiation, the exact same wild-type cell line. And I think you can appreciate that the picture immediately looks quite different. So we noted that there were a lot of other cells, probably much smaller cells, which were negative for both transgelin and calponin, indicating that this was probably a differentiation that had gone wrong at some point. So it's kind of, I think, nice to show that even if you have quite a lot of experience with this model, you sometimes do get dud differentiations like this. And it's quite important to make sure that you monitor your differentiations, and you do look at markers to make sure that you can eliminate differentiations like these from your critical experiments. I would also like to highlight the fact that there can be a lot of variability between wild-type iPS lines. So we did some bulk mRNA sequencing of our in vitro derived vascular smooth muscle cells from the 3 different lineages. So we generated lateral mesoderm cells, neurocrest cells, and paraxial mesoderm vascular smooth muscle cells. And these are this is just a heat map of some of the kind of classic housekeeping genes that you would use, for example, for your qPCR experiments. So you have things like HPRT1, GAPTH. And I think you can visually see quite immediately that there is a lot of variability in the expression of these housekeeping genes across the different cell lines, and also very clearly between the lineages. So this really does highlight that even if you have control lines, wild-type lines, there are quite a lot of differences between them that you should be aware of when you're designing your experiments. So then how do we tackle issues of reproducibility? I think the problem comes from the fact that when you want to generate iPS-derived vascular smooth muscle cells, it's very much a multi-step process. So each step that you take, if there is some variability, these things can then compound and add up to something where it results in quite a big difference in the final product that you actually make at the end. So I think there are a couple of ways in which we can tackle how we can get more reliable cells using this kind of system. So the easiest one, of course, is having very clearly written protocols. Another thing that we've noticed is, especially when it comes to training collaborators, for example, having in-person training is really invaluable because being able to show somebody through a microscope what the cells should look like at a particular stage of differentiation is much easier than months and weeks of emailing back and forth with lots of pictures. So that's really something that's quite important. The other thing that should be done is to define kind of clear quality control metrics at each stage of differentiation. So I showed you earlier the staining of the SMC markers, and you can see that there can be quite a lot of differences you can pick up as well. I think it's also quite important to do routine testing and checking for genetic abnormalities because, of course, stem cells are notorious for accumulating kind of pro-survival mutations, which may also affect your end result if you're not careful. And of course, we need to, as wherever we can and wherever possible and convenient, increase the number of replicates that we use in our experiments, as well as using multiple clones of the same line and using more lines just to try to get a better picture of what it should look like. It should also be noted that for rare diseases like Loeys-Dietz syndrome, like vascular Ehlers-Danlos, this might be more tricky to get many patient lines. But the system is very amenable to CRISPR-Cas9, so you could do some very clever knock-in experiments where you knock in patient mutations as well. So that's something that's quite valuable. And the other things that are maybe a bit harder to implement, but still I think worth thinking about, is the elimination of non-chemically defined reagents, such as fetal bovine serum. And our whole differentiation pipeline from iPS cell to the vascular smooth cell is all chemically defined, apart from the very end where we typically mature the cells in media containing fetal bovine serum. And this is something that is potentially an issue because if the batch of your FBS changes, or if you have a different batch to collaborators, that might have a knock-on effect on the kind of final product that you then assay. And of course, the last thing, which maybe is a bit kind of science fiction for us right now, perhaps, is perhaps automating differentiations using robotics. So currently, if you have one postdoc with technical support, I think you can just about manage to differentiate about 10 iPS lines. But if you were thinking of experiments in the future where you may wish to differentiate 100 lines, for example, then that's going to be very difficult for one person to manage. So this is kind of where you may wish to think about robotics and how you can remove the kind of human bias of the differentiations to make them go more smoothly, more reproducibly. And then lastly, I'd like to talk about how expensive iPS research is as well. So I had a brainstorm of how much we might spend in just 1 month of one person differentiating, let's say 2 or 3 lines in quite a dedicated manner. So I thought about the kind of costs of cell culture media, of the coatings and the different reagents you might need, as well as the kind of plasticware and other miscellaneous items. And this comes down to about 647 pounds, which as of this morning is $750 US. And this is, I think, quite a hefty price tag. And this is the cost of just simply generating the cells. This does not include the cost of any downstream assays, sequencing, or any drugs you may wish to use. And it's something that's really worth considering if you are thinking of going into iPS research. And something else that is worth highlighting is that, of course, the Sinha lab, we are a stem cell lab. So a lot of these reagents we buy in bulk. And we have a lot of recipes for kind of in-house formulations for medias and things to be able to save money. And so I imagine that if you were to buy commercial media as well, the price tag of this kind of research would go up by quite a considerable amount as well. So to conclude my talk, we went over the advantages of iPS models. It's of course a model of human disease. It's patient-specific. You can do some very interesting lineage-specific studies, as well as having a system that's amenable to CRISPR gene editing. And there are a number of applications that's possible using the system, such as drug screening, as I've shown you, as well as some really wonderful 3D modeling that other groups have done. Some of the drawbacks of iPS models, of course, is the fact that you have issues with reproducibility and variability. You also have a very steep learning curve. And of course, as you just saw there, the cost of iPS research is also quite high. And I think these 3 things together means that it's not something to be taken lightly, that it's really kind of a whole lab endeavor to be able to do iPS research well, because it does take a lot of input to to be able to get to a point where things run smoothly. And so then for future work and outlook, I think there are a number of things which we now think about doing with IPS models. First of all, how different patient lines respond to drug treatment. I've just shown you a little bit of pilot data that we have with regards to how different that can be in, let's say, GSK3 beta inhibition. But then there's also a lot of work that can be done in identifying the role of other cell types in the aorta, such as the endothelium, which has not been as studied as the role of vascular smooth muscle cells. And of course, this system can then be expanded to disease modeling in other aortopathies, as well as the kind of non-syndromic diseases that are perhaps less well studied. And I think the way we approach human disease is very much going from a mouse model, finding some drugs, finding something that seems to work in the mouse, and then seeing how human patients respond to that drug treatment. And I hope to have demonstrated to you today that there is a lot of value in adding a parallel arm of research with iPS cells and iPS-derived vascular smooth muscle cells, because they really can complement a lot of in vivo studies, in my opinion, because you can go through different testing of different drugs, different combinations of drugs, much more quickly using stem cells than you can when you design, wonderful in vivo experiments. And lastly, if you've enjoyed my talk today, and if you're interested in learning more, myself and colleagues have written a review paper relatively recently where we talk about a lot of the topics that I've touched upon in this talk in a bit more detail. So I do encourage you to have a look if you're interested. With that, I'd like to thank the British Heart Foundation for funding this work, the Sinha Group for their help and support, as well as our collaborators. And thank you very much for your time.
Hongorzul, thank you. That was really great. We've got some questions coming in. I encourage other people to type in questions for Hongorzul and also for our next speaker. But I think we'll go straight on to the next talk, and then we'll have time at the end for questions from and answers from both speakers. So Dogukan, can I get you to upload your talk and present?
All right, can you see my screen now?
Yeah, we can see that.
Great. Well, thank you for the introduction, Dr. Sinha, and thanks to the organizers for the invitation to talk. So I'll talk about our iPSC modeling studies of Loeys-Dietz syndrome to highlight some of the opportunities for iPSC modeling in human thoracic aortic aneurysm research, as well as challenges that we are exploring and trying to overcome. So there are several challenges in iPSC modeling, ranging from clonal heterogeneity to the off-target effects of the gene editing methods, and many of which were beautifully summarized by Dr. Davaapil. But in this talk, I will talk about the challenges somewhat more specific to our human thoracic aortic aneurysm research. So brief intro. Human thoracic aorta can be subdivided into 4 regions based on anatomic locations: root, ascending aorta, arch, and descending aorta. Animal model studies demonstrated that smooth muscle cells in the aorta are a major determinant of aortic disease. And these studies also provided many insights into the developmental origins of aortic smooth muscle cells. Again, briefly, aortic smooth muscle cells have different heterogeneous embryonic origins depending on their spatial location. 2 examples: ascending aorta smooth muscle cells are derived from both neural crest stem cell, ACSC lineage, as well as cardiovascular progenitor cells in the second heart field. But while the aortic root smooth muscle cells are mainly derived from CPC lineage. The several forms of thoracic aortic aneurysm, TAA, also show regional specificity. For example, Marfan syndrome and Loeys-Dietz syndrome often cause root aneurysm, while the bicuspid aortic valve-associated aneurysms are often observed in the ascending aorta. Compared to abdominal aneurysm, thoracic TAA's genetic predisposition plays a dominant role in TAA, creating, I think, many opportunities for iPSC modeling. There's been more than 20 genes associated with TAA, and we can also categorize them into subgroups, including TGF-beta signaling genes, extracellular matrix organization genes, small muscle cell contraction genes, etc. And today I'll present data about a Loeys-Dietz syndrome associated gene, TGF-beta receptor 1. Again, briefly, Loeys-Dietz syndrome is a genetic disorder. It's characterized by aortic root aneurysm as well as widespread connective tissue disorders. LDS patients carry pathogenic variants in canonical TGF-beta signaling receptor, TGF-beta signaling genes, including TGF-beta transforming growth factor beta receptor 1. So we identified an LDS family with aortic root aneurysm and dissection. All family members with LDS carry a pathogenic variant in TGF-beta receptor 1 gene. This mutation causes a single amino acid change in the kinase domain of the protein. So given that root is mostly populated with CPC lineage small muscle cells, we asked whether this pathogenic variant causes defective small muscle cell differentiation in a lineage-specific manner, leading to the root aneurysm. So to answer this question, we used the iPSC model. We generated control iPSCs from individuals with no aortic abnormalities. Then we knock in the mutation. We made mutant iPSCs using CRISPR-Cas9 gene editing. And similarly, we generated iPSCs from patient PBMCs, peripheral blood mononuclear cells, and corrected the mutation using CRISPR-Cas9 gene editing. Then we differentiated these iPSCs to smooth muscle cells through their intermediate progenitors, cardiovascular progenitor cells and neurocrest stem cells. So among the challenges I want to highlight here is that there are several differentiation protocols to derive vascular small muscle cells. And these protocols rely on different induction methods, and they often yield a non-uniform cell population with not fully characterized cell maturity. There's also a growing number of single-cell RNA sequencing studies profiling different small muscle cells from different regions of aorta, including from control tissue. So I think it'd be great to generate a reference cell atlas of region-specific small muscle cells using all this human data for several reasons, but also to map our iPSC-derived small muscle cells from different protocols and to optimize these differentiation protocols to improve their physiological relevance. So going back to the data, we differentiated our iPSCs through their intermediate progenitors, CPC and NCSC lineage. And first, we looked at the contractile gene expression in our isogenic controls as well as mutant, knock-in mutants. We found that the mutant cells had showed reduced expression of small muscle cell markers, including in the heterozygotes. But surprisingly, small muscle marker expression was not significantly different in NCSC lineage small muscle cells. Similarly, we corrected the mutation in the patient-corrected cell lines, which we call as I-wild type, had higher expression of these small muscle cell markers compared to the patient small muscle cells, and improved function as well. So I'll quickly go through the data to get to the challenges. We also did single-cell RNA sequencing profiling of these cells, which shows that ECM organization and small muscle cell contraction gene sets are enriched in the control clusters, whereas the mutant clusters were enriched in Wnt signaling and Rho GTPase signaling gene sets. And one key difference we observed between the mutant and control cell lines was cell proliferation. Many of the cell proliferation markers, upper right quadrant here, were enriched in the mutant cell lines. In fact, DNA replication and mitotic cell cycle gene sets were the among the top enriched gene sets in the mutant condition, and then we validated this higher proliferative activity in the mutant cells by EDU stainings. We also looked at the downstream signaling defects caused by this pathogenic variant. We assayed the phosphorylation status of many TGF-beta receptor 1 downstream effectors. Briefly, SMAD3 and AKT levels were reduced in the mutant cells, suggesting that these cascades, SMAD3 and AKT cascades, may be playing important roles in mediating the cellular response to TGF-beta receptor 1 activation. To sum up the data, we have reduced SMAD signaling, we have impaired AKT phosphorylation and higher cell proliferation in the mutant cells. So we picked two agents, activin A and rapamycin, to rescue the phenotype in mutant cells in vitro. Activin A signals through activin receptors and induces SMAD2/3 phosphorylation, whereas rapamycin is a known inhibitor of aortic small muscle cell proliferation. It also promotes AKT phosphorylation. So we treated our cells with either one of these agents or both agents, either during the progenitor cell to small muscle cell differentiation phase, which we called as early treatment, or after small muscle cell differentiation phase, which we called as late treatment. So both treatments increased the small muscle cell expression in the mutant cells. We also looked at the contractile function, and these treatments also improve the contractile function in the mutant cells. So going back to the challenges, in the study I presented so far, we were largely limited to two-dimensional monolayer culture. So I think although this model is reliable to understand the signaling defects caused by a pathogenic variant in a cell signaling receptor, the model lacks relevant environmental factors such as hemodynamic stress or intercellular communication defined by the vascular geometry. So next we aim to produce implantable three-dimensional constructs with relevant cues to make a more predictive human cell-based disease model. To do this, by the way, this is work done by Ying and AHA postdoctor, a lot of work done by Ying and AHA postdoctoral fellow in the lab. We seeded isogenic control and heterozygote mutant patient-corrected small muscle cells on PGA mesh. We culture them for 8 weeks. And at the end of the culture period, the control vascular grafts, which we called as BVGs, had robust mechanical properties with burst pressure values higher than 1000 millimeter mercury. Excuse me. Then to compare the in vivo performance of each pair of control and mutant vascular grafts, we implanted them bilaterally into the carotid, common carotid arteries of immunodeficient, immunocompromised nude rats. Then we monitor the in vivo patency, blood flow, mid-vessel inner diameter change of these vascular grafts bi-weekly using ultrasonography. These ultrasounds reveal that the patient vascular grafts the inner diameter of the patient vascular grafts gradually increased over time, starting at around 2 millimeters with a nearly 40% increase by week 8 post-operation. Of note, we repeated these experiments in the knock-in background as well, and we show similar results as well. Then to understand the molecular events underlying this dilation phenotype that we observe in the patient vascular grafts, we perform spatial transcriptome profiling of both patient vascular grafts and patient-corrected I-wild-type vascular grafts at week 8 post-operation. The resulting clusters showed differential distribution between different samples, but I would like to highlight 2 clusters here, one of which is the C2 cluster enriched in a smooth muscle cell marker, taglin, transgelin, SM22 alpha, and the other cluster, C3 cluster, enriched in many collagen-modifying enzymes, including PLUD1 and LOX. So these 2 clusters, these 2 more smooth muscle cell-related clusters, form the wall of vascular grafts, but they only labeled isolated regions in the patient vascular grafts. So then we did some gene set enrichment analyses again. The patient vascular grafts were enriched in injury and repair related gene sets, while the I-wild-type patient-corrected vascular grafts were enriched in ECM organization, collagen formation and modifying enzymes, and elastic fiber formation gene sets, which I think consistent with the stronger mechanical performance of these I-wild-type vascular grafts in vivo. A couple things I want to highlight here is that the TGF-beta signaling is still higher in the control in the patient-corrected vascular grafts compared to the patient, similar to the 2D monolayer findings, confirming the persistent defects caused by this mutation in vivo. However, we don't see one of the tough cellular phenotypes that we observed in the 2D culture was cell proliferation, but there is very little cell proliferation at this stage of the vascular grafts, and we don't see that phenotype here. So there are some also stage-specific phenotypes that we start observing. Another example I can give you is that we also did stainings to validate these collagen defects that we observe, and there is a higher density of thick collagen fibers in the patient-corrected vascular grafts at week 8 post-operation. Surprisingly, we do not observe this phenotype in pre-implantation vascular grafts. So making the vascular graft is not sufficient. Making a vascular graft in vitro is not sufficient to see this phenotype. We need to put these grafts in the animal, expose them to all those factors, including hemodynamic stress, to capture some of these phenotypes. So another challenge that I would like to highlight here is elastin. As you all know, elastin fragmentation is a hallmark of end-stage aneurysmal tissue. In our 2D monolayer culture model, we have very low to no elastin expression. Surprisingly, at 8 weeks post-operation, we start seeing some elastin expression in our vascular grafts. However, don't get too excited because we actually do not see any good elastic fibers that you would normally see in aortic tissue. So we are still far from getting any elastin in our vascular grafts, but I consider this as both a challenge and an opportunity as well, because perhaps we could use these grafts to better understand elastin regulation as well as elastic fiber formation. All right, so I'm going to summarize the data. We identified a family with a pathogenic variant and TGF-beta receptor 1. This mutation, this variant, preferentially affects CPC lineage smooth muscle cells, which could help us explain the aortic aneurysm observed in the LDS patients in this LDS family. We came up with a combination treatment with activin and rapamycin, which rescues the defects caused by the variant in vitro. The third conclusion I have is that I think our bioengineered vascular graft experiments show that the carotid arteries or common carotid arteries of nude rats can support bilateral xenotransplants of vascular grafts, and the mutant vascular grafts show dilation at 8 weeks post operation. And the last point I would like to make is that I think this model could represent a more physiologically, this vascular graft model could represent a more physiologically relevant iPSC disease model, and maybe could pave the way, help us understand early, mid, and late stage molecular events that lead to aneurysm formation. And in the case of this particular TGF-beta1 mutation, we see widespread collagen defects in patient vascular grafts. With that, I would like to thank Dr. Yang and Dr. Chen for their guidance, and Ying, some of our work I presented today, Han Dong, who did the original study, and the rest of the group, and our funding sources. And I'll be happy to take any questions at the Q&A session now. Thank you.
Dr. Mizrak, thank you very much for that excellent presentation. I'm going to open the floor to questions and encourage any of the audience members to type questions into the Q&A, and I can then throw that to our panelists. We've got one question already, which basically says, asking about viral vectors. What kind of viral vectors do you use to deliver the iPS transcription factors? If it's an integrating virus, could integration into a critical genomic area actually impact your results? Perhaps both of you. So maybe Dogukan, how do you make your iPS cells, and which viral vectors do you use?
We actually use plasmids of the 4 factors as well as BCL. I mean, I'd be happy to share the protocol with them, but yeah, so we use just plasmid electroporation, basically. Nucleoporation, yeah.
Great. And Dr. Davaapil?
So from our side, we use Sendai virus version 2, and this is a non-integrating virus, and it just induces the expression of the Yamanaka factors. And then afterwards, when we are ready to use the iPS cells, we check for the transgene as to whether it's still there or not before we use them.
Okay, great. I mean, while I'm waiting for other questions to come in, I have a few questions actually, which I mean, I thought these are both excellent talks that really highlighted some of the potential and really clearly showed the challenges of this kind of work. So a few questions actually. Maybe so, Dr. Mizrak, you talked about comparing either knocking in the mutation into a wild type line or taking the patient line. In your experience, have you found significant differences when you're knocking in the same mutation that you have a patient line for? Do you find that the different genetic background has an effect on the phenotype that you're seeing?
Absolutely. So not only different genetic, even for the control lines, if you knock in the same mutation, I see differences. I mean, I looked at this as both as bulk approaches as well as single-cell RNA sequencing type approaches. So I do see definitely some background effects, and we like to do it both ways because in the case, the way we do it, we both do the knock-in as well as patient-patient correction. This also cancels out any possibility of seeing any off-target, the phenotypes that are caused by the off-target effects of the gene editing methods. But overall, it's not just the knock-in phenotype. I think I do see major phenotype differences between different control lines when we differentiate them using the same protocol. So this is not too surprising, because if you take any human tissue, any male human tissue, for example, extracted or removed from the individuals from the same age, you will still need to use some data integrations tools to kind of overlap them. And the same is true for iPSC differentiation as well, I think.
Sure. Dr.
If I
Davaapil,
can, and Sanjay, if I
sure.
can add to your question, Dogukan's leading our lab, right? They're doing a great job. And when CRISPR-Cas9 become available and matured, everything, we do not need the patient's cells anymore. We're just knocking any mutation one when we study the alien disease. I don't think we're there yet. I don't think that's a reality. As regarding your question and regarding the patient's cell line and the wild type was knocking, are there any difference or not? We do see difference.
Mm-hmm.
And we can say the patients actually cell line and their phenotype is more severe than the knocking mutation. Just put it that way. Yeah, they're not exactly the same. You're knocking this mutation, you think you can mimic the exact same patient's phenotype. Some degree, yes, we can, but not 100%. That's individual differences. Now, when we start Loeys-Dietz project, and before Dogukan came in, we have the whole family, like 6 of them. We have another 6 controls. We try to compare just normal control to patients, the phenotype of the LDS cells, but because of the variation among the individuals, among the controls, among the patients, so it's very difficult to get to see the significant difference of true biology. However, the CRISPR-Cas9 give us the opportunity, give us isogenetic control, which helped a lot for our for the stem cell research. That's why we had a breakthrough, and we found out a really significant change between the mutation and the isogenic wild type background. It's a patient correction.
Sure, and we found some similar sort of findings to yourself as well. I'm going to ask about both of the presenters showed work on how the IPS model can be used to look at response to drugs, for instance. So maybe for Dr. Davaapil, how do you think this sort of work would integrate with informing clinical care? What do you see as a sort of pathway or route to that? Is it something that we could help clinicians?
Absolutely. I think iPS research in particular is very amenable to personalized medicine and really tailoring the drugs that you might wish to use to treat patients, because I think a problem that was encountered with the losartan studies was you just had some patients which responded really well to it and others that didn't respond to it as well as the others. And because fibrillin, for example, is such a huge gene, and any number of mutations there can cause a different effect, or any differences in genetic background can have a really strong effect, as you've also mentioned, that I think it's going to be a really important facet for personalized medicine.
Yeah. Dr. Mizrak, do you think, just a comment about using the system for, how do you see it for using the system for informing treatments, therapies for patient care?
I think, it's a complicated question because it really depends on what stage we are talking about, because a treatment that could be relevant for a 2D monolayer culture, it may or may not be relevant in a more advanced iPSC model. So it really depends on the phenotype, because based on what I see here, it starts as in the case of, for example, LDS study, it starts off as a small muscle cell phenotype, and then it evolves into a more extracellular matrix phenotype over time. So it really depends on what stage we are talking about and what stage of the disease we are talking about. But overall, I think one thing that we'll have to consider with any drug screen studies is the cost, because it's not cheap to make vascular grafts. So rather than a screen, I would probably do a more targeted approach. First, figure out what the phenotype is, and then come up with a more targeted strategy.
Great.
Okay, I think the iPSC screening always give us a like target drugs, right? That could be some clue, some candidates for later on for animal model treatment, and that's I think that's a good start. What do you think, Sanjay?
Yeah, I mean, I think there are a lot of possibilities here. I mean, both Dogukan and Hongorzul raised some good points. I think there is potential for drug screening, as we've shown, but I think that the more complex studies that you've shown with the grafts can only be used in a small number of drugs in a more targeted manner. So I think that we've got a spectrum here. We can do quite high throughput screens with these, but we have to be able to funnel that down into really picking the key new therapies that we might want to take to clinical trials, and those, I think, is where your grafts then come in. So we've got different opportunities for different aspects of that pathway. I've got a question from Joe Velchev, who thanks you for the talk, Dr. Mizrak. He says, why did you prefer the CPC smooth muscle cells in your experiments? Also, how relevant is the graft if you only have one cell type? What about endothelial cells?
Yeah, so the reason we selected CPC lineage small muscle cells, because the root is mainly populated with those cells, and we see a lineage specific defect caused by this variant affecting CPC lineage small muscle cells. In terms of the second part of the question, yes, we are doing a monolineage analysis. We are essentially just seeding one particular small muscle cell type on our vascular, and we make the vascular grafts from a monolineage cell. So in reality, the aorta is far more complicated than that. Not just endothelial cells, we also have fibroblasts, and furthermore, there is also different subtypes of potentially of small muscle cells. And depending on the area, you probably have multiple small muscle cell lineages, right? Including if you want to mimic the ascending aorta. We are not there yet. In terms of endothelial cells, though, sometimes you can also observe endothelialization of coming from the host over time when you keep the vascular grafts in the animal long enough. But the bottom line is that in the future, it'd be great to make these vascular grafts from multiple cell types as well as multiple small muscle cell lineages.
Yeah, there's a follow-up question from DT Shetty about the grafts, which is, DT comments that there's quite a bit of variability between your different replicates. How many grafts would you need as a gold standard before you can draw conclusions?
Yeah, so we have 6 replicates, and basically the data I showed you for spatial transcriptomic, which is quite an expensive assay, we did 2, but we did all the validations in. You have to do so those are gene expression results. When you do the orthogonal validations in many replicates, which in our case is minimum 5 to 6, that would help us draw conclusions from the data.
Back to the first question about endothelial cells. So based on all the studies we showed, our animal study and other studies, smooth muscle cells is the main contributor to the aneurysm, to the aorta. You knock out the specific, smooth muscle specific gene, like a receptor 1 mutation knockout or knock-in mutation, the mouse develop aneurysms, right? And we know the smooth muscle cells, the key player for the aortic, for the aorta. We cut the aorta out. Majority is smooth muscle cells, and the thin layer intima is endothelial cells. I do not think endothelials play a huge role in the development, but current knowledge didn't show that endothelial cells play a huge role in the aneurysm formation. That's why we started with the smooth muscle cells. So I think that's a key player. And then also I mentioned that, and fibroblasts could be another player, and endothelial cells could be a player too. And then they crosstalk all the time. And you know, large mutation, and you knock it out in with the innocent background, knockout background, you develop aneurysm. So that's cross-talking too. So for this specific model, yeah, I mean, we started with the muscular vessel first, and it helps actually because the endothelial cells from the host animal can populate the vessel, prevent thrombosis, which we do not see thrombosis in our vessels. So yeah, I think it's a cell type we should pay attention to. I mean, we just started this. I think that's why we're focused on smooth muscle cells.
I think starting with smooth muscle cells is the right place to start, but clearly there's potential to refine this even further. But very exciting systems, and I think we'll stop there. I want to thank both the speakers for great talks, particularly showing not just the potential, but how sort of practical issues to do, and really explain that clearly to the audience. I want to thank the Marfan Foundation and GenTAC Alliance for helping us to set up the webinar. Thank you all.